Welcome again students of Prep 3. يلا يا شباب نكمل مع بعض منهج الساينس الترم الأول. عندنا يا شباب يونت 4 ده جزء خاص بالبيولوجي. عندنا الجزء ده لذيذ جدا. Number 1 أو lesson 1 living organisms can be classified into two types. Number 1 unicellular living organisms. Number 2 multicellular living organisms. Unicellular means their bodies consists of one cell, for example, amoeba, paramecium, euglena, and bacteria. Multicellular, they are living organisms where their bodies consists of several cells, for example, plants, animals, and humans. Types of cells: somatic cells and reproductive cells. All body cells called somatic cells except reproductive. For example, your body contains muscles, hair, skin, kidney, heart, and so on. These cells called somatic cells. Also in plant, cells of root, stem, leaves, and so on called somatic cells. But the other type of cells is reproductive cells, as testes in animal male, ovary in animal female, and also anther in plant male and ovary in plant female. These cells consists of organelles. The most important organelle in the cell is nucleus. Nucleus contains thread-like bodies called chromosomes. Chromosomes, they are thread-like bodies present in cell nuclei, and they represent the genetic material of living organisms. Again, somatic cells and reproductive cells contains nucleus. Nucleus is the most important organelle in cell. Also, nucleus contains thread-like bodies called chromosomes. Chromosomes represent the genetic material of a living organism. Chromosomes consists of two threads, each one called chromatide, and they are connected in a point in the middle of them called centromere. Centromere, it is a point of connection of the two chromatides of chromosome. Again, each chromosome consists of two threads connected at the center, each thread called chromatide, and the point at the center called centromere. Centromere, it is a point of connection of the two chromatides of chromosome. Chromatide also consists of two threads, each one called DNA, means nucleic acid, and protein. DNA and protein. DNA is a nucleic acid that carries the genetic traits of living organisms. Okay? The somatic cells, its nucleus contain in human 46 chromosomes or 23 pairs, so it called diploid number or 2n. Also reproductive cells, its nucleus also contains the same number of somatic 46 chromosomes or 23 pairs, so it called diploid number or 2n. But gametes, which produced from reproduction or reproductive cells, contains n only or haploid number or have number of chromosomes in somatic and reproductive. So gametes contain 23 chromosomes only. Okay, we have a problem. If the number of chromosomes in animal pancreatic cell is 16 chromosomes, what is the number of chromosomes in the following cells? Number 1, skin. Skin also somatic as pancreatic, so it contains 16 chromosomes. 2, sperm or ova. Sperm or ova called gametes. Gametes contain n or haploid number of chromosomes, so it contains the half number of pancreatic. means 8 chromosomes. Number 3, fertilized ovum. Fertilized ovum means ovum after fertilization by sperm, so it equals the somatic and reproductive cells, so it contains 16 chromosomes. Chromosomes consists of 2 chromatides connected at centromere, and also each chromatide consists of nucleic acid called DNA and protein. Chromosome chemically consists of not chromatide and centromere but nucleic acid, DNA and protein. Chemically, chromosomes chemically consists of DNA and protein. But if we have chromosomes, generally consists of space and space. Chromosomes generally consists of chromatides and centromere. The number of chromosomes in the living organism is not from one species to another, but its space in the members of the same species. Again, the number of chromosomes in living organisms differs from one species to another, but it fixed in the members of the same species. How? In human, male, female, child, adult contain 46 chromosomes, so the number of chromosomes in each of them is fixed. Importance of chromosomes. Number 1, they represent the genetic material of living organisms. 2, they have the main role in cell division. 3, knowing the number of chromosomes helps in identifying species. Now we will study the cell division. It's a complicated process through which the living cells divides into 2 cells or more to aim growth or reproduction. We have 2 types of cell division. The first cell division called mitosis and the second meiosis. Mitosis occurs in somatic cells. Meiosis occurs in reproductive cells. The aim of mitosis division is growth or compensating dead cells. But the aim of meiosis division, which occurs in reproductive, is formation of gametes. Mitosis, represented by 1 cell to n, divides to give 2 similar cells, each of them to n. Meiosis, represented by 1 cell to n, divides to give 2 similar cells n plus n, and also each of them divides to gives 2 new cells, each of them also n. Now we will study the mitotic cell division, which occurs in somatic cells in human body, except we have 2 types of cells can't be divided at all. Number 1, nerve cells. Why? Because it doesn't contain centrosome. Number 2, adult red blood cells. Adult red blood cells. Why? Because it doesn't contain nucleus. It doesn't contain nucleus. So nerve cells and adult red blood cells can't be divided at all. Importance of mitotic cell division. Number 1, growth. Number 2, compensate damaged cells. Number 3, completing asexual reproduction, which occurs in plant and microorganisms. Before starting mitotic cell division, the cell passes through a phase called interphase. During interphase, the cell is prepared for cell division by DNA is duplicated and appear in form of reticulum, and occurrence of some important biological process. So we have An important give reason question. Interphase has an important role before starting mitosis. Why? Because in interphase or during interphase, DNA duplicate. The first phase in mitosis, number one, prophase. Prophase. Number one in prophase, chromatin reticulum condenses, then appear in form of chromosomes as long and thin threads. At the end of this phase, the nuclear membrane and the nucleolus disappear. Number three, in animal cells, a network of fibers called spindle fibers appear from centrosome, but in plant cell, spindle fibers formed from condensing cytoplasm. Why? Because plant cell doesn't contain centrosome. Because plant cell doesn't contain centrosome. So, we have two words, centrosome and centromere. Centrosome, organelle in the cell, which form spindle fibers in prophase. But centromere, it is a point of connection of the two chromatides of chromosome. We have very important give reason question, the difference in the origin of spindle fibers in animal cell than in plant cell. We can answer that by number three. In animal cells, fibers called spindle fibers appear from centrosome, while in plant cell, spindle fibers formed from condensing of cytoplasm. Number two, phase number two, metaphase. In metaphase, the chromosomes are arranged at the cell center of the cell or cell equator, which are connected with spindle fibers at the two poles of the cell. Number three, anaphase. Anaphase, number one, the centromere splits or divides lengthwise into two halves, the chromatides in each chromosome separated. Number two, spindle fibers shrink and two identical groups of chromosomes are formed at each pole. Number three, each group of chromosomes migrates towards one of the cell pole, as shown in the figure. Phase number four, telophase. Telophase, nuclear membranes and the nucleolus are formed again. Number two, spindle fibers disappear. Number three, at the end of this phase, the cell divides into two identical or similar cells where each nucleus has a complete number of chromosomes, 2n or 46 chromosomes as in human. We also have an important question give reason. The change which occurs in telophase are called adverse changes. Again, the changes which occurs in telophase are called adverse changes. Why? Because it's adverse to the changes that occurs in prophase. It's adverse to changes that occurs in prophase. We have very important note, liver transplantation. Liver transplantation. Liver cells don't divide in the normal conditions, but when the liver injured or cut, they retain the ability to divide the mitosis, but under certain conditions, to compensate the lost part. When a person suffers from liver failure, doctors can take a liver loop from a donor and transplant it in sick person. The liver of the donor grows back into the normal size. If the person who receives the transplant accepts the new liver, it can also divide and grow, replace the damaged one. So this process called liver transplantation.